Hello everyone, welcome you all to the week 11 NPTEL TA session on personalized therapy, which is a part of genome editing and genetic engineering. Okay, and this is Subhasmita from IIT Indore. I am a PhD scholar here, and as a part of my PMRF TA duty, I am teaching in NPTEL session. Okay, so let's start our today's session, or we have waited so long for other people to join. Please let me know if my screen is not visible to you or if I am not audible to you. Okay, so first thing, what is personalized therapy? Okay, first we need to know about it, then we can do the questions, right? Here we are having this assignment session because it will help you to do your assignments weekly. Okay, you can go through your videos and weekly lecture notes and this assignment sessions also, so that it can help you understand the lessons properly, and it will help you to do your assignments, and I guess it will be fruitful for everyone. Okay, so moving to the uh, theory part where we are right now, this personalized therapy is called generally one size does not fit all. Okay, I just took this slide, everything from your lecture notes only. You can go for that also. So, personalized or precision medicine therapy refers to medicines or therapy that combine genetic information of a person with phenotypic and environmental characteristics to produce healthcare tailored to the individual and eliminates the constraints. Okay, so what happens generally? uh, there are lots of people in this world, and there are different genetic variation is present, right? So if we give one medicine to each and every person, then we can't say that it will work for everyone. So to get proper result for every person, and to get the and to get this disease cured for everyone, every patient, we need to go for this personalized therapy, where we combine this genetic information of a particular person or patient with phenotypic and environmental characteristics. Like if the person is staying in the upper altitude region or high altitude region, or if the person is staying in a plain area or it in a seashore area like a local coastal area. Okay, that's also depends upon their genetic variation and uh, change of medication. Okay, so for example, today nowadays we go for boutiques for preparation or making of different kind of designers uh, clothes for ourselves. So like that we can go for personalized medicines also, or which is specifically designed for a particular person with their phenotypic and genotypic characteristics. Okay, that's why it is called one size does not fit all. That means one medicine cannot be assured to be good for everyone. Okay, so generally, drugs are tested on a wide group of people, and the average reaction is considered. And this type of evidence-based medicine, that is medicinal or medical decision making based on empirical data, is based on the rule of averages, whereas personalized medicine understand that no two patients are similar. Now you understand the difference between them. So one can ask the question here, so what is the difference? Every drugs go through different types of clinical phase 1, phase 2, phase 3 clinical experiments, right? So what is this need for this personalized therapy? So here is our answer, that generally for when drugs get tested, they use or they refer to a group of people. Okay, wide group of people. Then they collect this empirical data, and then they average it, mean it. Okay, but therapy, we assure that, or we think that no two persons are similar. So we can't go for this average data for everyone. Okay, I guess this is the difference between personalized therapy and general therapy. Okay, so here in the right side figure, you can see here, just a minute. So here you can see this is the single nucleotide polymorphism which is seen in different persons. So see here, there are three different persons of three different individuals are present, and there is this genetic variation is present, which is called, and it is just a simple example to show you how these persons are being different via a single nucleotide polymorphism. This is called polymorphism because it is seen in number of patients. Poly means a large number of people, and now you can see that it can be adenine, guanine, or thymine for different persons. Okay, so this single nucleotide polymorphism changes the characteristics, changes the genotypic and phenotypic behavior of these three individuals. Okay, so one of the key causes of this disparity is that people inherit different gene variants, little variation of which can have significant impact on how the body reacts to a specific treatment. Even a little difference, such as a single nucleotide base being misspelled, might have significant clinical effects. So, we are studying genome editing and engineering, and this is our week 11 session, and about 10 session has been passed, and we studied a lot of about genetic variation and how genetic engineering can help us to cure different types of diseases. So from that point of view, you can understand till now that there is different types of genetic variation is there, and if a single nucleotide can changes, then that also can changes the person's behavior, person's everything about a person, from its genotype to in a protein level or RNA level or anything. So if the protein becomes different, then functions also become different, right? So like that. So the single nucleotide polymorphism are crucial in determining a person's susceptibility to certain diseases and drug response. Okay, so this is what we need to understand about personalized therapy before starting it. So this is just a kind of introduction. Okay, so let's go to the question session here. Ayurveda classifies all individuals into different prakriti based on the theory of Vata, Pitta, Kapha or Tridoso. Which one is it? Here we will be talking about Ayurveda also. This is the kind of fun part here. We'll be talking about our traditional medicinal therapies, which is, I guess, everyone need to know about it, right? What, how it is started and how our ancient uh, scientists do this. So, please try to answer this question in 2 minutes. Then we'll be discussing the whole thing. Everyone try to answer this question. Okay, let's talk about it. So Ayurveda, everyone knows about Ayurveda, right? I guess in Indian household, everything is there in your kitchen, and you understand that Ayurveda is so helpful in during this COVID era, right? For our natural immunity system. So every household, mothers told us to drink this haldi doodh, that is turmeric milk, and ginger, and different types of natural ingredients which helped us and boost our memory, sorry, immunity. Okay, so Ayurveda is a natural healthcare system that dates back more than 5,000 years in India, and it thinks that each individual is unique and has a particular constitution, and hence emphasizes on personalized treatment. Okay, so this is the motto of Ayurveda, which believes or emphasizes on personalized treatment. And today our topic is personalized therapy. That's why we are more interested in studying Ayurveda here. And it classifies all individuals into different prakriti, okay, based on the theory of tridosa. This is independent of racial, ethnic, or geographical consideration, and may provide appropriate means of classifying phenotypes to be considered collectively for genotyping. Understood? So how this is classified, you can see here. There are three types of things which Ayurveda generally believes that human body is made up of space, air, fire, water, and earth. This is generally called Panchatattva. Okay, you will find out this word in Ayurveda specifically called Panchatattva. That means five essential components of life, and these are space, air, fire, water, and earth. So that exist in human body as the three primary humors known as tridoso. So how this is called tridoso? So it is three things, that is vata, pitta, and kapha. So what are they? So vata includes qualities reflecting the elements of space and air. Okay, if we combine space and air, that is called vata. Pitta includes elements of fire and water. If we combine fire and water, it is called pitta. Kapha qualities reflecting the elements of water and earth. Okay, so the components of space and air combine to form the vata dosa. Pitta dosa is made up of the elements fire and water. Finally, kapha dosa is derived from earth and water components. Okay, so Ayurveda believes that every person has their different qualities or different constitution of this vata, pitta, and kapha. Constitution means you can say every person has different this varieties, or how many percentage it is. Okay, so our answer here is tridoso. Fine. So this is called prakriti. Generally we can say it in English as nature. What type of nature an individual has. I guess this is the general idea of Ayurveda and the different tridosas we are talking about. So let's talk about another question. Ayurveda categorizes drugs according to Rasa Panchika, that is Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug as follows. Find the wrong combination. Rasa means taste, guna, effect, virya, potency, vipaka, post digestive state. Try to read this question again and again and answer this. You can take 2 minutes, then we will discuss about it. Okay, then let's discuss about it. So earlier we have been talking about this categorization of Tridoso, right? Tridoso includes Vata, Pitta, and Kapha, and these three things has different proportions in case of different individuals. So that's why Ayurveda generally categorizes persons or individuals based on their uh, Tridoso qualities or Tridoso amounts, and then it suggests drugs according to or personalized to that particular individual. Okay, so similarly Ayurveda categorizes drugs according to Rasa Panchaka, that is Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors represent in the drug, namely rasa, that is taste, guna means property, virya means potency, and vipaka means post digestion taste, and prabhava means effect. Okay, whereas modern pharmacology attributes the drug action to the chemical structure of a molecule. Fine. So generally, in Ayurveda, we or Panchaka. Pancha means five. Okay. So generally in Ayurveda, they consider that if a drug is going to act in particular individual's body, then what are the different qualities it must have? So it includes rasa means taste. What is the taste of this drug? Guna means property. So what is its property? Property, it may includes different types of physical property and chemical property, like your melting point of the drug or any other effects or any other chemical structures or physical characteristics it has. Okay. Virya means potency, how much it can affect your body. Vipaka, post digestive test. Post digestive test means in after digestion, how is it going to act in your body. And prabhava means effect, what is its effect, how it will affect the individual, how it will change, if it is affecting it in a has any side effects, or it will act normal without any side effect, like that. So these are the five things what Ayurveda considers for a drug to be classified as better or good. But whereas modern pharmacology believes or generally characterize this molecule or drug based on their structure, chemical structure, which ring it has, which benzene ring or phenol ring or which type of ring is there. So how is it going to act and what are different types of chemical reactions going to happen. Okay. So this Rasa Panchaka modality can deliver treatment because it considers the person's prakriti. So earlier we have talked about prakriti is based on, or prakriti means nature. Nature is based on tridoso qualities, as well as the pharmacodynamics and pharmacokinetic properties of a drug, as opposed to modern treatment which elicit different responses from person to person even when using the same drug for the same disease. Now you are trying getting to understand, I guess, because we have been talking about personalized therapy. That's why the tridoso qualities of, if two patients are different, then the drug efficiency or effect of the drug is also different. That's called pharmacodynamics. Okay. So for so what is it saying here, that which elicits different responses from person to person when using the same drug for the same disease. Okay, so here is our answer. Guna. Rasa, taste. Guna means potency. Guna means what? Your property. And virya means potency. Vipaka means post digestive taste. So Garrod is best known for his book Inborn Errors of Metabolism, where he claimed that four diseases Alkaptonuria, albinism, cystinuria, and pantosuria were inherited as Mendelian autosomal recessive trait, and it was published in which year? Here we are talking about little bit of history. That is, if it is 1909, 1902, 1809, or 1990. You can guess if you don't know the answer. Okay, let's talk about it. So in 1902, Garrett published the paper The Incidence of Alkaptonuria, a study in chemical individuality, where he suggested that alkaptonuria is not the manifestation of a disease but is rather of the nature of an alternative course of metabolism. Okay, so basically, you must have heard about it for the example of autosomal recessive trait. Okay, and he is best known for his book Inborn Errors of Metabolism, published in 1909. So here is your answer. Inborn Errors of Metabolism, that is published in 1909, where he claimed that four diseases, that is alkaptonuria, albinism, cystinuria, and pentasuria, were inherited as Mendelian autosomal recessive trait. This foresightful effort paved the way for the study of hereditary illness and established Carrer's status as the father of medical genetics. So now you can remember it, or not, it's up to you, but he is the father of medical genetics. Okay, so here is their answer, that is 1909. Human Genome Project completed in which year? 1998, 2001, 2010, or 2015? I guess uh, everyone heard about this Human Genome Project. So please try to answer it. This happened in modern era only, so that's why we need to know. Okay, okay. So now you got your answer right. I guess everyone answered correctly. That is 2001. Let's talk about it. So this Human Genome Project is completed in 2001 and opened the door for to better knowledge of precision medicine. So here you can see how this flow of time in this flow of time medicine or personalized medicine has been evolved. Okay, so in 1977 Sanger method, so Sanger method is generally used for DNA sequencing. I guess everyone heard about it and we have also read in our sessions. Okay, so Sanger method generally used for DNA sequencing, and it was discovered in 1977. Okay, next is human mitochondrial genome sequence, that was in 1981, and human genome project started from 1990. And 1995 complete cell genome, 6 eukaryotic genome, 2001 complete human genome project. Then second generation sequencer analyzer 2, research human microbiome project, third generation nanospore sequencing, and now we are at a third stage of human microbiome project. Okay, so in this course of time you can see how this personalized medicine has been evolving from 1977 to 2019. Okay, so let's move to our next question. So Sanders group published the first proof of concept that next generation sequencing technology may be used to diagnose genetic diseases in September 2009. After a few months, they reported Miller syndrome, the first recessive disorder. So, in that case, they have used whole exome sequencing, whole genome sequencing, or whole proteome sequencing, or whole plasmid sequencing. Please try to answer this question, then we'll be discussing it in 2 minutes. Great, everyone answered correctly. That is whole exome sequencing. Okay, so let's talk about it. What is genome? What is exome? Then introns and proteome, plasmid. What is it? Okay. Yes, yes. So this is the process of next generation sequencing, where you can see here the first step is, so this is a schematic representation. You can go for the molecular level representation in your lecture slides only. So see here, first step of NGS, next generation sequencing, it is library preparation. So this genome is now fragmented, and then adapter was added to make a DNA library. Next, DNA library bridge amplification using different types of amplification cycle. This is also called this Illumina method of DNA sequencing. Okay, you can find out it. So next is DNA library sequencing. So here you can see different types of computational methods have been used, and it has been done on a chip. Okay, and here it shows your data. Then after analyzing the data and alignment, you can find out the assembled sequence. You can, if you remember this shotgun sequencing of DNA, shotgun method of DNA sequencing, then I guess you will find similarities between them. So what is this shotgun method? Generally, in that method, the scientists or researchers cut the DNA, or make the DNA fragmented. After that, they try to patch it off along depending upon their sequence variability. So that's why, and that's how they get the assembled sequence. And from the assembled sequence, you can find out how it is sequenced, okay, using Illumina method. So that's why it is called next generation sequencing. And Sanders group published the first proof of concept of NGS technology used to diagnose genetic diseases, which was in September 2009. After few months of, they reported Miller syndrome through whole exome sequencing. So what is whole exome sequencing? Everyone knows about introns and exons, right? So every genome. So this first thing you need to know about the central dogma of life. What is central dogma? Generally in molecular biology, we can say that central dogma means DNA replicates itself to form the next DNA, right? And DNA go through the mechanism of transcription to produce mRNA, right? Then after mRNA is produced, it moves through post transcriptional modification. So in that post transcriptional modification, there is two methods generally happens. What is it? One is splicing, another one is methylation chain addition. Okay, so what is the splicing mechanism? In that splicing mechanism, the non-coding part, okay, the non-coding part of the gene is been spliced out. So this non-coding part is called intron, and the coding part of the gene is called exon. So that's why introns has been removed and only the coded part of the gene remains there. And from that spliced mRNA, translation happens, and that leads to production of protein using ribosomes and other enzymes. Okay, so this is the central dogma of life. And now this Miller syndrome was reported using the whole exome sequencing. That means they have used the coding part of the gene only, not the non-coding part. But nowadays scientists have found out the different specification and functions of this non-coding part of the DNA, right? So you may find different papers on research papers on non-coding RNA, non-coding regions which has specified in specific functions also, right? So we'll talk about it more if we get a session according regarding this. So let's move to our next question. Engineering cells for precision medicine is called precision immunotherapy, precision immunization, immunity, or personalized immunization. Please try to answer this question. Today's topic is personalized therapy. That's why we are talking about different types of personalized methods and precision medicines. Okay. Others, please try to answer this question. I got one answer only. Okay, then let's discuss about it. So, engineering cells for precision medicine is called precision immunization. So, how this happens? Precision immunization in the human immune system protects the body against external infections and eliminates endogenously damaged cells to keep it healthy, right? So in the last several decades, vaccination has become a common kind of healthcare, continuing century-long attempts to harness immune system for human health. So how this personalized or precision vaccines work, you can see a schematic diagram here. So this is kind of natural infection, and these are the disease outcomes. What is includes fever, nausea, hospitalization, and other. So natural infection, disease resistance, vaccinated. So so this vaccination can be specific to particular group of people. Like now we have been gone through this pandemic era where COVID-19 vaccine has been given to each and every group of population, right? First it was developed for, or it was given to a particular age group, that is between 40 to 60 range or above 60. Okay, then it was become the age group becomes about 20 to 40 range, and then children. Okay, so why they made this division of age groups? Because the immune system of different individual is different, or in a different age group, right? You also agree on that point. Because in a person with 60 plus age, okay, and if we compare that person's immunity to a adult person, then there might be a quite big variation we can see. So that's why this vaccination also depends upon that particular things, and this includes antigens and adjuvants. So what are adjuvants? Adjuvants are substances which enhances our antigenic property, which helps the antigens to express more, or express in a proper way, so that antibodies can be formed inside the body. Okay, so and this includes the formulation development, including hydrogels, cellular vehicle, viral vector, nanoparticle, nanocarrier, and microparticle. So there were two types of vaccines we got in our COVID-19 era. So can you tell me which viral vector was used for the vaccine? There are two types of vaccines, right? So which viral vector was used for the vaccine purpose? Can anyone tell me which viral vector was used for Covishield? Two vaccines were there, that is Covaxin and Covishield. Is it? So, okay, then let me tell then. So, it was the adenovirus vector. Okay, remember it, because COVID vaccines, and how, because we are biologists and we are reading about genetic engineering, so we need to know and we need to remember these things, right? So they use the adenoviral vector for designing of the Covishield vaccine. So, and this delivery route includes injection, transdermal catch, or can be sprayable gel. Fine. So how they get toolkit for building vaccine to mimic natural immunity? So how they get built? So see here, three age groups are there: adult or aged group or children group. So this system biology, that is omics, age specific human in vitro modeling, and this leads to preclinical application before clinical testing of three phases. That can be monkeys, apes. So rats are the first phase. We cannot go to monkeys or apes directly. We can go for rat. First it is in vitro level. Then we need to go for this mouse model. We have already been talked about this different types of mouse models in our previous session. So generally mice is used because the genomic sequence is similar to us. So that's why we can go for mouse, then apes and monkeys. Okay. So that's how this precision vaccines we design. So you can say that what is the example of precision vaccine? Then Covishield and Covaxin are one example of precision vaccines, because they are differently designed for different age groups. The composition and the immunity strength and the adjuvants, antigen, everything is different for different age group. Okay. We cannot give one vaccine to particular person looking at the genotypic and phenotypic calculation, but we can divide them as per the age group. Fine. I guess everyone is clear with precision immunization. Okay, let's talk about it more. Here is our next question. Engineering smart biomaterials for drug delivery systems that can detect the demands of a person's physiological condition and change their drug's release profile accordingly is an appealing method for creating precision immunization, precision pharmacokinetics, pluripotent stem cells, precision biomaterials. Read the question twice or thrice, then try to answer it. Okay, I got answers A and B. We have just talked about precision immunization, right? Do you have any doubt regarding this? See here, engineering cells for precision medicine is called precision immunization. But here we are talking about engineering smart biomaterials for drug delivery system. So how can these two be same? Just try to understand, okay? See here, engineering cell for precision medicine. Here we are engineering cells. That's why it is called precision immunization. But now here we are seeing engineering smart biomaterials for drug delivery systems that can detect the demands of person's physiological condition and change their drug release profile accordingly is an appealing method. So why we are talking about this drug delivery system and drug release method? See, there are different types of delivery systems are present. Okay, here in this figure it is present only, including hydrogels. Even you also know that some drugs or medicines come as liquids. For example, your cough syrups. Some are like tablets or capsules. There are two types of capsules are different, tablets are different. Okay, don't be confused between them. So you can see different types of capsules are present, tablets are present. So in our case, in our daily to daily life, we can see that there are different types of drug delivery methods. So like that, cellular vehicles, viral vector, we have just talked about our Covishield vaccine, microparticle, nanocarrier. Okay, there are different methods of formulation drug development. Okay, so that's why we need to deliver this completely authorized or developed drug to the patient's body, right? Let's talk about it more. So engineering smart biomaterial for drug delivery system is appealing method for creating precision pharmacokinetics, right? So he's here, you see here in the right side figure you can see determinants of inter-individual variation in drug response, then knowledge of aging effect on pharmacokinetic and pharmacodynamics, knowledge of pharmacogenetics can avoid adverse drug reactions including hypersensitivity. Okay, and liquid biopsies mRNA measures including cytochrome P50 also helps. So this all everything lead to precision medicine for older persons. Oh, this is one example of particular age group medicine design. But this smart biomaterial, why we need them? Specifically, we can say that after developing a drug we need to deliver it perfectly, right? We have just used our so much effort in developing a particular drug. So in that, if the delivery method is not correct, for example, if you ev- everyone knows that the drug dissolves in our blood and blood circulates the drug particles throughout whole body. Okay, so no one is a kid here, so everyone knows like Disprin or some kind of medicines which are specifically for headache, they directly don't go to the head. So they first diffuses out in the blood, then it circulates it each molecules inside the whole body. Okay, so for that also, and there are specific medication. For example, we are trying to give a specific medicine for kidney or kidney disease or liver disease or any particular disease, and for that we also need specific type of delivery system, because sometimes what happens in case of some patients, the drug delivery system is not good, that's why the drug doesn't affect that particular person. Okay, so this kind of thing includes totally overall can be called as precision pharmacokinetics. Now you got your answer. Okay, here is our next question. Dash are one class of smart polymers that can repair their structure after repeated injury and are frequently injectable by needles. Polymeric biomaterial, miRNA polymer, precision biomaterial, self-healing polymer. Earlier we have been talking about pharmacokinetics, right? Precision pharmacokinetics, where we need or develop certain kinds of different biomaterials. So here we have been classified these biomaterials into different classes. So now you need to characterize which one of the class of smart polymers can repair their structure after repeated injury. Okay, let's discuss about it. I got two correct answer, that is self-healing polymers. Okay, three correct answer, four correct answer. This is called self-healing polymer. So smart polymers are one type of soft materials that respond to environmental changes, and the thermally sensitive polymers, which are frequently employed as cell carriers and in 3D printing, are can be good example. So self-healing polymers are one class of smart polymers that can repair their structure after repeated injury and are frequently injectable by needles. Another type of material with a capacity to remember its original shape is safe memory polymers. So now we got to know about two types of polymer, that is self-healing polymer and safe memory polymer. Okay, so this is kind of reminds you back about your regeneration process, which is found in case of human liver and lizard tail. Okay, so if you don't know, or if you know, let me tell you this. This human liver, if some part of the liver damages out, then it can regenerate itself. Okay, second thing, lizard tail also. If it gets cut off, then it can generate a new tail. So this is a kind of regeneration process. So from that point of view, these materials, these smart polymers has been developed which can heal itself, or it can, these are called smart polymers because they respond to different environmental changes, and if they get torn or cut off or something like that, then they can be regenerated. So that's why it is called self healing polymer. Okay, so these smart polymers can transport. Why we are using this, that's also we need to know, right? That they can transport proteins, drugs, or cells. They can be used in precision medicine, minimally invasive surgery, and bioprinting, thanks to the injectability and safe memory capabilities. Okay, so specifically these smart polymers can be, you must have heard about this nanomaterials, nanotubes, nano carriers. Nano means very very small. Okay, so nano carriers, nanos, for this drug delivery system. So that's like that smart polymers also can be used to transport different proteins, drugs, and cells, and specifically they can be used in minimally invasive surgery also. Okay, and we'll be talking about different types of uses of this in your upcoming slides. So let's talk about it more. Here is our next question. The term theranostics, which was first used by John Funkhauser, refers to scientific advancements that aim to create more specialized and personalized treatment for a variety of organoid condition, animal model condition, pathological condition, or humanoid condition. Read this question again and try to answer this. Okay, let's talk about it. So first thing, what is the term theranostics? Theranostics means combination of therapeutics and diagnostics. So first we need to understand the terms. therapeutics and diagnostics. What is diagnostic? Diagnostic means how to check or how to know that this person has this disease. Okay, so that is called diagnosis. That means we need to identify a certain problem in a patient or individual. Therapeutic means medication, how we can treat this disease for this particular individual. Okay, so if we combine diagnostic and therapeutics, then it is called theranostics. And the term which was first used by John Funkhauser refers to scientific advancements that aim to create more specialized and personalized treatment for a variety of pathological conditions. These advancements also aim to combine diagnostic and therapeutic applications into a single agent, resulting in a promising therapeutic paradigm that includes diagnosis, drug delivery, and treatment response monitoring. So it provides a diagnostic therapy for specific patient, testing them for potential adverse drug reactions, and then customizing a course of action for them depending on the test results. Okay, so if you can go through different types of uh, campaigns like this. For example, if you if you find different types of your skincare products or skincare sites, you will find out that first, or in case of different types of sites where they mm, give you ayurvedic medications or some type of skincare or hair fall routine or something like that. So they will first ask you what is your hair type or what is the hair length, how if it is dry or it is humid, or if your hair freezes or not. So these are the kind of symptoms they are trying to analyze what is the problem. So this is a part of diagnosis. Okay, so when they provide you this oil or this routine you have to follow, this food or this vitamin you have to take, then that is the therapeutic part. So this theranostic approach has been used for used by different commercial products or different kinds of commercial sites also nowadays. Okay, so this includes diagnostic therapy for specific patients. So why it is specific patient? Because this is personalized therapy or precision medicine, because every person has different problems and different genetic variation, and testing them for potential adverse drug reaction. Okay, some people have specific allergies to some kind of products also. And then customizing, everyone knows the meaning of customizing. That means we are trying to customize a particular thing according to our need. Okay, and a course of action for them depending on the test results. So this is a variety of pathological condition, which pathological condition the person has. Okay, so this theranostic approach is quite new in this modern era, and everyone is using for personalized medicines. Fine, so let's talk about it more, and moving to our next question. A high throughput drug testing platform. Earlier we have been talking about this testing of drugs, right? It provides a diagnostic therapy for specific patient, testing them for potential adverse drug reaction. So how we can test them? A high throughput drug testing platform for precision medicine may be constructed using the which technique, which combines organoid cultivation and organoid behavior monitoring into one device. Try to answer this question. If it is tissue on chip, cells on chip, organoid on chip, or organs on chip, which method it is? Okay, I got your answer. Let's talk about it. First, we need to understand what is this method, then you can answer it properly. Okay, so single organ chips. That's why it is called organs on chip, not organoid. Why it is called organs, not organoids? Because you can see here, this is a chip and there are four wells. And these four wells can act as four different organs. Like one well is for heart, other is for your intestine or digestive system, third one is here for lungs, and fourth one is for your brain. Okay, so these four wells can work as different organs. Okay, that's why it is called organs on chips. Organ level physiology and functionality, disease modeling, toxicological assignment. So before, earlier we have been using, and still now also, this organs on chip is a quite advanced method and it is not available commercially for everyone. Okay, so for till now today also scientists use animal models for testing of different types of drugs and medications. These animal models include mice or zebrafish or any kind of different types of your pigs or apes or whatever it is. Okay, different types of animal models has been used, but here uh, and for that purpose lot of mice has been killed, which is against of our ethical rules. Okay, and for that purpose also, and it is there are quite side effects and different types of quite hard to handle and different types of limitations are there. Okay, so for that this one chip organs has been developed by scientists. So this is called organs on chip, like four different organs can be can be present on a single chip, and it includes tissue-tissue interface, biomechanical motion, multiple cell type, fluid exchange, permeable barrier, vascularization or innervation. So here you can see how these chips can be interconnected. Okay, here they have been showing different types of modeling, including personalized medicines, which we are interested in, or drug screening, or systemic modeling of diseases. Okay, this is all for organoid organs on chip method. Here we are specifically using a particular tissue. Okay, tissue culture. Now you understand it more. It is a particular tissue culture from a particular organoid. That's different organs can be placed one chip, and this middle line can be act as your blood vessel. Fine. So this is provides the vascularization and connection to different organs, like as as it is in our whole human body. So these organoids have been produced in vitro using biopsies. Okay, biopsy, everyone knows that is some tissue is been extracted from the patient tumor or like that, from cancers of the liver, gastrointestinal tract, head or neck, pancreas, prostate or etc. One of the most important aspects of correctly anticipating a drug's effectiveness is preserving the heterogeneity of the tumor microenvironment. Earlier we have been talked about tumor microenvironment in a lot more detail. If you have not attended that session, you can go for the sessions which is present in our YouTube videos. You can go for that week 10 session or week 9 session. We will find out how tumor microenvironment heterogeneity helps the tumors to grow. Okay, so, a high throughput drug testing platform for precision medicine may be constructed using organs on a chip technique, which combines organoid cultivation and organoid behavior monitoring into one device. Fine. So that's why it is called organs on chip, because multiple organs can be played on a one chip. So here answer is organs on chip. Okay, so I guess everyone is clear. So this is all for today. If you have any question, then you can ask me. We have talked about personalized therapy from ancient to modern era. From ancient era, we have seen that this Ayurvedic Panchatattva and Tridoso and Prakriti of individuals, how it is affecting personalized medicine and Ayurvedic pharmacokinetics. Then we have seen that, and we discussed about use of genetic engineering in personalized medicine while helping precision immunization, precision pharmacokinetics, and then we move to this theranostic part, and then our organs on chip method. So this 10 question overally summarizes your week 11 lecture series, which has been talked about personalized therapy. If you have any question regarding this, you can ask me. Okay, then if you have no question, then we can end this session here. Thank you all for joining.